This is Campbell's Biology, Chapter 16, Molecular Basis of Inheritance. This is Part 1. So we're looking at the history of DNA, the discovery, its structure, and so on. So the famous pair, Watson and Crick, 1953, they built a model here. They basically got to play with a toy, and for doing so, they won the Nobel Prize. Of course, it was more involved than that. DNA is super popular. Very important to lots of our studies now. And we know it's the hereditary information, but we didn't always know that it was the hereditary information. Early 20th century, we had to balance. We thought maybe it was protein. I guess I shouldn't say we. I wasn't around then. It was potentially protein. A large majority of the population thought that was the genetic information. So the first experiments that kind of led us down this line were done by Frederick Griffith with pneumonia, and he worked with two strains, a pathogenic strain and a non-pathogenic strain. The pathogenic strain was the S cells, or smooth Streptococcus pneumonia, because they were protecting themselves from the animal's defense system. And the R strain, the rough, was non-pathogenic. They didn't kill the animal because they didn't have that capsule around them, and thus the immune system was able to kill it off. So when he injected S cells, the mouse died. When he injected R cells, the mouse was healthy. Then he decided to try and warm up the S cells and kill them, and then he injected them into the mouse. Well, when he did so, the mouse stayed alive. It didn't die. Then the key here was he took heat-killed S cells and he mixed them with living R cells, and when he did so, the mouse died. And this was the first basis, the first evidence that transformation, a term that he coined, existed, where something from the heat-killed S cells passed into the R cells and thus killed the mouse. Again, so he called this transformation, which is basically the assimilation of external DNA into a cell. Then there was information that came from bacteriophages, which infect bacteria, and they were used by Hershey and Chase, and Hershey and Chase used them to show that DNA was a genetic material, and they used a T2 bacteriophage. So what they did is they took the phage here, these little guys, and they labeled, in some of them, they labeled the sulfur with radioactive sulfur, S35. And sulfur was already known at the time to only be incorporated into protein. And then they took a separate batch and they radioactively labeled the phosphorus, P32, which was known to only be incorporated into DNA and not protein. Then they allowed them to infect bacterial cells, and they tried to identify which part fluoresced. And when they took the protein-labeled ones, what they discovered after centrifuging down post-infection was that the pellet at the bottom with all the bacteria was not fluorescing, was not radioactive, but the supernatant, the fluid on top, was. And therefore, the bacterial cells didn't get the radioactive sulfur. When they did it here with the radioactive DNA, they found that it was all in the pellet in the bacterial cells, so therefore they showed that the genetic material, the thing that the bacteriophage was injecting, was putting into the cell, the bacterial cell, was DNA. Prior to the 1950s, we already knew sort of a basic structure of DNA, or let's say the things that were involved, that being a nitrogenous base, a sugar, and a phosphate group. So here you see a nitrogenous base. This happens to be thymine, or T. Here's the sugar. It's a 5-carbon sugar, and here's the phosphate group, PO4. So additional evidence that DNA was genetic material was from Chargaff, who analyzed DNA and basically determined across different species that DNA varies, the composition varies, and thus, again, that diversity enables lots of things to be different, making it a credible candidate. So then came Rosalind Franklin, who worked in Maurice Wilkins' lab in the UK, and Rosalind Franklin was actually a physicist, and she was a specialist in X-ray crystallography, where you take a solid crystal and you shine X-rays through it, and you expose a photographic film on the other side based on how the light refracts or bends, and based on that you can make interpretations. Don't ask me how. This picture told them that DNA was a double helix and the exact structure and so on. Pretty amazing stuff. But Rosalind Franklin took this picture, and there's some controversy here because Rosalind Franklin had suggested that she didn't give permission to Watson and Crick, who actually used this picture. Watson and Crick claim that they got the picture from Wilkins, who was a friend of theirs, even though Rosalind Franklin never published, and in fact, as a result, Franklin was never mentioned in the Nobel Prize. So Watson and Crick used that X-ray crystallographic picture to identify this, and this is the model that they built, of course. They decided it was two anti-parallel sugar phosphate backbones, and the bases were in the middle. And the bases alwa' paired with the same things. Adenine always went with thymine, and cytosine went with guanine. Here is that structure. You'll see later we're going to talk about 5' and 3'. I want to identify here that DNA is anti-parallel. This double helix, one end of this strand is the 5' end, and the other end is the 3' end. And the other strand is the inverse, or the opposite. If this is the 3' end, this is the 5' end of the other strand, and this is the 3' end of that. They are anti-parallel. They go opposite directions. So Watson and Crick then hypothesized that adenine and thymine formed 2 bonds between them, and cytosine and guanine formed 3 bonds. And this was part of their specificity of pairing, that A always went with T and C always went with G. And here you see that A and T had 2 hydrogen bonds. The pink dots represent hydrogen bonds. And here we have 3 between G and C. And we'll take a break on this one.